Okay, guess we'll get started. I'm James. I'm a postdoc in the Rose Lab at UCSD, and I'm going to be talking about the genetic history of a human specialist mosquito, Aedes aegypti. So the Rose Lab primarily studies Aedes aegypti. It's a problem mosquito. It's responsible for yellow fever, dengue, Zika, chikungunya. It's an urban specialist, and it really loves feeding on humans. And we're pretty confident that this species originated from Africa, spread to the Americas via the slave trade, and then with the increase of urbanization and globalization, spread everywhere else. And I'm just putting up these specific locations because I'll come back to this as we have samples from those areas. But this species isn't strictly a human specialist. There's evidence for another ecotype, which exhibits more generalist feeding behavior, actually preferring other animals over humans. And these two ecotypes diverge in their environmental niche. So the human specialist appears more often in urban areas, and the generalist is found more often in forest environments. When you look at genetic ancestry across the global distribution of Aedes aegypti, you see multiple genetic lineages. We primarily just see one outside of Africa, and then three prominent ancestry groups in Africa. And then when you look at the feeding preference of those samples with the host choice assay, so this is just mosquitoes picking between a human and a non-human odor, we find this correlation between genetic ancestry and host preference. So this index just goes from 1, all mosquitoes from this population preferred went towards the human hand, to -1, all mosquitoes from this population went to the guinea pig. So we see this association, and the red ancestry here looks like human specialist ancestry, and the blue ancestry looks like generalist ancestry. So a question that we're investigating in the lab is, where in Africa did the human specialist ancestry originate? Where and how? And from previous work in the lab using coalescent analyses, it was estimated that the divergence between these two ancestries occurred roughly 5,000 years ago at the end of the African humid period. This was a big transition, especially in the Sahel region. All of a sudden you have these 9-month dry seasons. And it's theorized that the resulting scarcity of the natural tree hole habitat because of these long dry seasons forced Aedes aegypti to rely on artificial containers near human settlements to breed. Okay, so also previously, my PI, Noah Rose, he observed that the human preference behavior seemed to be correlated with precipitation seasonality. You can think of that as the difference between the driest and the wettest months, with more human specialist behavior at the places that had this higher precipitation seasonality. And then, so the Sahel region has high precipitation seasonality, and several of our human specialist populations, which are the redder dots here, pop up in Sahel. So Sahel seems like a possible origin, but first we needed more data from the actual region to check, and then first check for this association again between ancestry and human preference. So this was the original data set comparing host preference with human specialist ancestry, and you can see we're missing a lot of, like human host preferring populations. We only have 3 here. So by adding in more Sahel populations, we fill this in a bit. the association still holds. That's great. We can also leverage other samples for which we only have ancestry, not behavior, to increase the number of populations and try to figure out like what's driving the presence of this human specialist form. So specifically now looking at ancestry as the predictor, as the response variable, when we compare model fits with bioclimate variables, human density, farming practices, we find that farming practice is actually the best predictor of human specialist ancestry, with specifically agropastoral millet farming. So I'm just coding this as like 1, 0. This is either the dominant practice or not. That comes out as explaining the variation the best. And this practice is prominent in much of Sahel. So that's the maroon line here is this agropastoral. And so the theory is that the pots used for water storage by millet farmers are acting as a breeding site for Aedes here. And you might have noticed as I flashed through this figure before with all our new data, it's okay if you didn't, that there are lots of points that fall outside of our best fit line, suggesting that genome-wide ancestry might not be the best predictor for host preference. Instead, maybe a particular region of the genome is. And so we do see these regions of the genome in gray here that seem to be more associated with host preference, and also where there's this elevated shared derived variation between some of the African human specialist populations and the out of Africa populations, which are super human specialists. So when we just isolate those regions in gray and do the same stuff I showed on the previous slides, we find the proportion of human specialist ancestry at those regions shows a stronger relationship with host preference than the genome-wide proportion. And then when we go back and look at all the environmental variables that predict human specialist ancestry at the outlier regions, we find, a model with agropastoral still comes out on the top, but it's now with this human density parameter. So together, these two parameters were the best predictor of human specialist ancestry at the outlier regions of the genome. Okay, that's all super cool, or at least I think that's super cool. We have an idea of what's driving contemporary distribution of human specialist ancestry, but it would be great to also try to figure out which populations in Africa have the older human specialist ancestry to investigate the Sahel origin hypothesis further. So what we do, to do this, we first want to isolate just the human specialist ancestry, which is a somewhat of a difficult problem in admixed populations, because we don't want the blue ancestry mucking up our downstream analyses. But using phased genomes and local ancestry inference tools, we can isolate that human specialist ancestry. So the next few slides are just looking at these, just looking at the tracks of human specialist ancestry. We can check for a signature of founder effects within the human specialist ancestry. And so the alternative hypothesis to a Sahel origin in the literature so far at least has been Angola. And this is justified because Angola is the likely point of migration from Africa to the rest of the world. So we're kind of comparing Sahel origin versus Angolan here. When we use tree mix to create a maximum likelihood tree, the Sahel population's in blue, so it's Ngoye and Chez. These show up as more basal, followed by Angola, and then Bangkok as expected. So that shows the expected signal if Sahel contains these founder populations. And then we can also do a similar thing, but we add a couple of outgroups, and we add the outgroups by first doing a reverse masking for a couple of populations. So we have like these 3 populations that are serving as outgroups, where we take only the generalist ancestry for those specific populations. And then we take the human specialist ancestry for the other populations, combine them all into a DXY tree, rooted by this related Aedes mascarensis. And again, we see evidence for these Sahel populations popping up as more basal to Angola. Okay, so just to summarize everything real quick, we find this association between human specialist ancestry and host preference. That holds when we add the Sahel populations. It gets stronger when we just look at the outlier regions. And then the distribution of human specialist ancestry can be explained by agropastoral millet farming practice and also human density. And then with the couple of analyses that I did at the end, I showed that Sahel populations appear to be more basal to Angola at least. Yeah. Okay, and I just want to thank the members of the Rose Lab and my collaborators and the funders. Yeah, Sean.
Just out of curiosity, can you tell us what proportion of the genome was associated with the human specialization?
Like what proportion has the human specialist ancestry?
Correct.
The maximum is like 0.3.
0.3% of the entire genome.
No, it would be 30%.
30%.
Yeah. Oh, that's the. Oh yeah. So I guess we have time for any more questions. Yeah.
Are you able to, like, the differences between the human specialist and the generalist? Is there like a specific PCR test or something like that you're able to tell, or do you have to go deeper looking at it?
We have to go deeper right now, but the hope is soon we'll develop an amplicon assay where we can tell the differences without sequencing so many loci. Yeah.
Do you think it's possible that human preferences evolve multiple times?
That's interesting.
I have no idea.
I mean, I haven't seen any evidence for that, I guess. The thing is that I guess it is like theoretically possible, but it doesn't seem to be the case that it evolved multiple times and then when you look at the genetic ancestry globally, you really just see this that red ancestry everywhere outside of Africa. And so if there were these different you would expect to see maybe a second or third lineage if this was happening multiple times, right?
Are there variation in host preference at all at the individual level within the ancestry grouping?
At the population level?
Yeah.
Yeah. Yeah. Thank you.
Hello, thank you all for coming. I'm going to present this study that Kira Delmore and I did. The punchline for the study itself is in the title, so you don't have to actually listen. But I think what's important is that we're using a methodology in a hypothesis testing framework that hasn't been done much in the past, and I think this study can serve as an example for how it might be used much more frequently in this manner in the future. What we're doing is taking genomic analyses, and as we know, we can use now well-covered genomes of single individual diploid animals to recover population demographics through their history. Long-term effective population sizes are within our grasp with readily obtainable genomic data sets. This main method, pairwise sequentially Markovian coalescent, or PSMC, was introduced by Heng Lee and Richard Durbin, well over a decade now in Nature, and some of you may remember those human demographic curves that they showed us in their Nature paper. I see we don't have a pointer. Those are human populations, and you can see that they were focused on the population bottleneck that we out of African humans share. But since then, they've been applied widely among non-model organisms, and what you see is that basically in this individual is representing a lineage history of effective population size going back in time from, on the left, close to the present 10,000 years in the past, all the way back to the right in a logarithmic scale to, in this case, 3 to 4 million years. You can see that estimates as you move toward the recent become, have much wider confidence intervals. So you have to take those more recent estimates with a big grain of salt. But as you move back in time, you can see those confidence intervals narrowing as that individual comes to represent more and more the actual lineage history. Well, what we've done, as I said, is use this methodology to test a hypothesis about the adoption of a life history trait and what that major life history trait's effects are on effective population size relative to members of the clade that don't have that life history trait. Here, the life history trait that we've chosen is obligate long-distance migration, and the clade of birds in which we're looking at them is the clade Catharus and its sister Hylocycla. And these are, as that photograph shows, a fairly nondescript forest bird. Omnivorous, breeding in the Americas, throughout North America and into northern South America. That clade is a pretty decent representation of the relationships among them. The birds that are shaded are sedentary. Those that are not shaded are obligate long-distance migrants. We made 3 predictions about the effects of migration on the long-term effective population sizes in the migratory birds relative to the sedentary lineages. The first was we predicted that effective population sizes would on average be higher among the migrants. We predicted that variation in those population sizes would also be higher. And we predicted that early population growth in those lineages would be higher because seasonal migration opens up both geographic and ecological space, thus achieving that first prediction. The reason behind these hypotheses is a combination of geography and ecology, and we really can't tease the two apart in this, but the example plate on the left shows a typical migrant lineage, Catharus minimus, breeding largely across northern North America, wintering in northern South America. The one on the left shows a typical resident lineage, and that's Catharus fuscator, breeding sedentarily in northern North America and middle America. The ecological space on the bird that moves a lot is obviously going to be a lot wider, potentially, than among the individuals of the sedentary lineages. And I've got the little maps of all of them on the right there, but you don't need to read those. Those 2 exemplars are pretty typical. I should mention that if you think of North America in geographic terms, it's roughly an inverted triangle, and so as you as a lineage or population breed farther north, there's bigger geographic space available to you, as well as a larger potential ecological niche. Well, this is a PSMC data set for Catharus minimus, and what we did is take that main PSC curve, chopped it off at 50,000 years. That's fairly conservative, but we wanted to eliminate the uncertainties that come with the more recent estimates of effective population size. And we've looked at these curves then from 50,000 years in the past back to the limits of the data, to the origins of the lineage. Those values are then averaged. Those are effective population size estimates by time interval. And then we took standard deviation and did a coefficient of variation for each of these data sets. And then we demarcated a trough and a peak for that early growth period. The time period between trough and peak we call delta T. That's the period of time over which they experience growth. And then the degree of that growth is basically how big is that peak in a manner that's relatively comparable among lineages. And then the rate of that growth, the degree of growth, basically the slope of growth through time. I want to throw in some caveats. The PSMC is not a silver bullet. Selection, inbreeding, and isolation can all 3 affect effective population size estimates. In our data set, it's probably population structure that's going to have the biggest effect. However, that effect is most likely to raise effective population size in the resident taxa. And also, as I mentioned before, its effects diminish backwards in time as the genomic record in the individual comes to represent more accurately the lineage history. And again, that's the same image I showed before, showing that uncertainty in the present, narrowing in uncertainty toward the earlier time periods. And those earlier time periods do actually become fairly important. We ran a bunch of these. We had 8 migratory lineages, 6 sedentary lineages. These are just some of the examples. I might mention, bioRxiv is a wonderful thing. I had all these data analyzed, the slides all made. A paper shows up on bioRxiv and says, oh, some of those peaks may be artifactual. And congratulations to the authors and to bioRxiv. Some of those peaks were artifactual. Those have all been corrected. Keep your eyes on bioRxiv so you don't give a mistake in your paper or your presentation. Results. None of our focal variables were correlated or significantly associated with the thrush phylogeny. To our surprise, there was no phylogenetic signal. This enabled us to use fairly simple non-parametric statistics to look at the major focal variables of the study. And these are the results which I'll walk through. As we predicted, the mean effective population size in migrants was larger than among residents. The coefficient of variation was larger among migrants than among residents, as we predicted again. And the degree of early growth was also higher among migrants, as we had predicted. However, our prediction that that would be accomplished by a rate of growth was wrong. That was not the case at all, and to our surprise, it was delta t, time between trough and peak, or the length of that growth period, that enabled the migrants to achieve a higher growth peak early on in their trajectories. However, when I say early on, the numbers of delta t there were rather shocking. So you'll see that the average delta t for migrants was about 3.1 million years, and among residents about 1.7 million years. Really much longer periods of time than I would have expected as a migratory bird biologist for these lineages to take advantage of new geographic and ecological space. Well, let's look a bit now. So that was our hypothesis testing. Now we're going to step over to using PSMC and its more traditional tradition. Can you have a tradition for a method that's 13 years old? Genomics moves fast. The traditional way in which PSMC uses is a descriptive and discovery science methodology. And that's what we're using here. We're saying, let's look more closely at those early growth periods. What I've done is taken the early growth periods, taken the peaks for all the taxons, put them at time zero. So we can look at all the growth periods of all the lineages together relative to each other. You can see the red ones are the migrants, the blue ones are the residents. And here you can see on average, yes, indeed, the red ones do grow to a taller peak, and they do grow over a longer period of time. But you can see that there's one resident that's acting very migrant-like, and there's another resident in there that's acting, or sorry, another migrant that's acting very resident-like. So this is, again, all on average. And there are some really weird exceptions in there that will bear out some interesting future investigation. But the other way in which PSMC has largely been used in the literature is to overlay multiple lineages in the same time frame. This is the actual time frame of all the curves, all the data. And what we notice right away is the heterogeneity. So what we'll frequently do in examining PSMC plots among lineages is look for concordant movements in those effective population sizes, basically often attributed to large climatic effects that have simultaneous influences among multiple lineages. These animals don't show that, which is really quite surprising. So it's surprising because they are all closely related and they occupy a really similar geographic space, not just in proximity in many cases, but indeed overlapping, especially with the migrants and the residents in winter. These initial growth periods, which I mentioned were remarkably long, parallel the expansion and adaptation phases of taxon cycles, which is a framework of lineage expansion and eventual contraction, which they also show through evolutionary time that encompasses both biogeography and evolutionary ecology. And this was not something we expected at all. It's simply noticing the parallels between two, what I would have thought, very disparate areas of investigation. Those of you who know about taxon cycles, E.O. Wilson, Bob Rickliff, Spiff Birmingham, Pepke et al., the theories developed in island systems, the studies have all been done in island systems. This is the first evidence I've seen that shows that there may be mainland parallels as well. This connection is tentative, but this framework really does bear further investigation. In conclusion of our study, our main predictions were borne out. On average, seasonal migrants have larger effective population sizes, greater long-term variation in those sizes, greater degree of initial population growth, although that's not achieved by rate, contrary to our expectation. And as I mentioned, those initial growth periods were very long, and they parallel in time and shape the expansion adaptation phases of taxon cycles. In sum, seasonal migration imbues these lineages with fundamentally different population size attributes through evolutionary time compared to their closely related resident lineages. Now I want to set our study results aside, cool as they are, and step back and comment a bit more on comparative paleodemographics. Despite PSMC's limitations, no method of demographic reconstruction is consistently and reliably accurate. But if we take a uniform application of powerful methods like this to genome scale data in closely related groups, we can obtain new insights into the evolutionary effects of major life history strategies. And with that, I'll thank the institutions and individuals who made this work possible, and I'd love to take any of your questions. Yes?
Would you expect interglacial cycles to have affected migratory birds more strongly than resident birds?
That-
And if you do, why do you not seek your evidence?
That's a great question. So you have to immediately decouple effective population sizes with census sizes. And as we can see from the post-glacial expanse from the last glacial maximum, the 10 to 20,000 years in the past, the absolutely phenomenally rapid expansion of several of these migratory lineages to occupy formerly glaciated regions of North America. NE just doesn't keep up with those rapid fluctuations. There are other aspects of PSMC, those time intervals also encompass, in many cases, multiple glacial interglacial cycles. So we're talking about rather long periods of time over which those rapid movements will become blurred. But over the long, again, as we predicted, despite that dampening of fluctuations, if you will, and the decoupling from census sizes, they do show higher variation. So they do show the effect, even though it's smoothed out. Other questions? Yes?
Thanks, Kevin. That was super interesting. I've asked it at another seminar, and I can't get my head around it. All the PSMC plots go back to like several million years ago, but a lot of these species, I don't know specifically about the thrushes, they're not that old. So how do you interpret the plot where it goes beyond the speciation?
Oh, that's fascinating. And I didn't have time to get into that, but those lineages and sisters actually usually meet where they were indeed the same population, and sometimes they overlap for a little period there. So, but that's a great question. Thank you.
All right, great news. Hey everyone, thanks so much for joining this afternoon. My name's Julia Kreiner. I recently started my lab at the University of Chicago, but I'm talking to you about some work I started in my postdoc at the University of British Columbia in collaboration with Lauren Reesberg and Stephen Wright at the University of Toronto. So today, that's going to be kind of evolutionary ecology motivated, but mostly focusing on some results that we can test with evolutionary genomic approaches. So I've been working on Amaranthus tuberculatus, also known as common water hemp or by farmers as pigweed, and developing it as a model system to study rapid adaptation. And that's because it's a really problematic agricultural weed that's become so on really recent timescales. And so it's really a species that thrived despite the conversion of its habitat to agriculture over just the last 200 years. And why it's so problematic is it's evolved widespread resistance to herbicides, to almost every chemical mode of herbicide control that we've been able to innovate. I'm not actually going to be talking about herbicide resistance or agricultural adaptation today, but something that I felt like I couldn't avoid working on the system, and that's that it's actually the diversity, both phenotypic and genomic, is structured by separate sexes in the species. So it's dioecious, which is pretty rare for plants. The ancestral form is hermaphroditic, and it's also wind-pollinated and diploid. And so we've done a lot of work looking at agricultural adaptation. This species is native to the Midwestern United States, so we have pairwise sampling of natural and agricultural populations. And we grow these out in a common garden. We can see sex really shapes most of the genetic variation and phenotypic variation across the landscape. In particular, I'll show two just fitness-related traits here, growth rate and flowering time. And what I was really interested to see is that, you know, of course, there's sexual dimorphism, there's differences in traits between males and females, but the amount of sexual dimorphism is also environment dependent. So we see bigger reaction norms or larger differences between sexes in agricultural compared to natural habitats. So I said I'm not going to be talking about agricultural adaptation, but differences between sexes might actually be involved in kind of this plant's invasion into agriculture. But why I think this is an interesting system to learn about the evolution of separate sexes is it's kind of rare to have this context of the evolution of separate sexes or dioecious in this ancestral, in this genus that's predominantly monoecious. So that means that individuals have both male and female flowers on the same plant. So while there's about 50 to 70 species, the majority which are monoecious, it seems like there's been multiple transitions to dioecious or separate sexes in the genus, in addition to what we see in tuberculatus. And so it's a nice system for testing two kind of contrasting hypotheses, evolutionary ecological hypotheses, about how separate sexes evolves in plants. So the canonical pathway is this two-step pathway where we move from the ancestral state of hermaphroditism, which includes monoecious, to dioecious. And so this takes two steps. The first step is the invasion of a hermaphroditic population with a male sterility mutation. And so that transitions the population to polymorphic for females and hermaphrodites when that mutation becomes homozygous in one individual. The second step is the invasion of a female sterility mutation. And when that occurs, you have its complete transition to dioecious. We then expect recombination suppression to evolve to prevent those sterility mutations from recombining onto the same background, which would be bad. But a less well-tested pathway to the evolution of separate sexes is this monoecious paradioecious pathway. And this is because it's pretty hard to find a background genus or family level that's predominantly monoecious. And we can imagine a monoecious plant, right, that has 50% male flowers, 50% female flowers. Well, to get to dioecious, complete separate sexes, you might actually have quantitative selection on sex allocation, where you might have much more continuous variation in investment into female or male flowers on a single individual. And so this is a less well-tested hypothesis that I wanted to see whether we have any support for in water hemp. So in order to do that, we have to learn and identify what the architecture of sex actually is, and whether that supports either of these two models. So one way we could ask that is whether there's evidence for recombination suppression in the species, what might be the mechanism for that, so we can use genomics to do that. Do we see evidence of a single kind of large effect locus as you would expect in a two-step model where recombination suppression has evolved? And does it completely explain kind of phenotypic sex? Lastly, we see a lot of phenotypic variation across males and females, and so does sex-related haplotypes also reflect that variation at the genomic level? So to test these questions, we developed some more genomic resources in the species, specifically comparative genomic resources, and this is just kind of a preliminary pass, but we performed really high-quality sequencing of 3 males from across our collections, and so these kind of spanned latitude ancestry, and those are highlighted by those yellow stars, and we used high-C, high-Phi, PacBio sequencing to produce really contiguous high-quality reference genomes. I'm not going to show you kind of those plots, but you'll just have to take my word on this, and I'm happy to chat about it later. And we combine that with previous population genomics data sets, so about 186 resequenced samples that we had grown in a common garden, that we had sexed, that we can combine to test them in these questions. So the first thing I wanted to do was to use our population genomic data to identify alleles associated with sex. And because we have haplotype phased assemblies from males that may capture, for example, an X or a Y, or if it was a ZW system, 2 Zs, we could actually leverage those separate haplotypes to perform a competitive mapping approach, where we combined both haplotypes into a single reference genome and map short read data to that. And that could allow us to distinguish mapping between closely related genes on the X or the Y, for example. And so when we do a GWAS, zooming in at a single chromosome, what we actually see is able to resolve a 4 megabase sex-linked region on haplotype 2 of chromosome 1, but not on haplotype 1 of chromosome 1. So this region actually contains about 103 annotated genes, including some really interesting candidates that I won't go into too much today. But for those plant biologists out in the audience, that includes things like FT, the flowering time gene, which was really exciting to see, and other things that have been functionally validated for female function, like the Madsbox transcription factor in rice. But we can overlay kind of this GWAS Manhattan plot with other population genomic inference to get a sense of what mechanisms and evolutionary processes might shape sex determinant in the system. And so one of the things we might expect if it was an XY system is to have an excess of heterozygosity in males compared to females. And so that's what I'm showing you here, overlaying that to the same signal, GWAS signals. And we can see almost exactly the same pattern for sex-associated SNPs that we do for this ratio of male to female heterozygosity. So what that implies is that these sex-linked SNPs on haplotype 2 of chromosome 1 are actually mapping to the Y, right? This is a region that's homozygous in females. We can then turn to kind of comparative genomic approaches and try and understand, well, why do we see such an extended region of linkage in this? And so we know that we're mapping to the reference here, haplotype 2, which we've identified to be the Y, and we can kind of compare. Let's see if I can pull up. Can you see a mouse? No. Okay, so we can compare kind of the synteny of genes on the X compared to the Y. And if it was a one-to-one rate relationship, that would mean that there's complete, the same orientation on the genes on the X and the Y. But that's not what we see. We see inversions, so flips in the orientation that surround kind of this sex-linked region. But really notably, between kind of 40 and 43, we see a complete lack of genes on the X compared to the Y in this region. So that's actually telling us that these sex-linked alleles seem to be mapping to a Y-specific region, but it's not actually that simple. And so when we remember, we actually have assemblies, haplotype phase assemblies from 3 separate males, and when we bring in those separate assemblies, what we see is there's not just differences in sequence content between the X and the Y, those 2 other Ys we've sequenced also have quite a lot of variation in how many genes are present in this region. So it's not necessarily Y-specific, but it's a polymorphic region for which these sex-linked alleles are mapping. I was really shocked to see that inversions, which are a really typical mechanism of recombination suppression in this region, don't overlap with any of these sex-linked SNPs. And so I really wanted to make sure that that was the case and we weren't missing anything with these kind of 3 comparative genomic samples. So what I'll show you is actually how enriched this sex chromosome is here for inversions. So we see about 12 inversions segregating just between these 6 haplotype phased assemblies. But when we then overlay that with population genomics, so we pull out those regions and we look at population structure just within those inverted regions. As you can see, there's strong population structure, as you would expect from an inverted haplotype. So there's kind of 3 different possible genotypes in this region, but sex does not load on any of the 12 inversions. I'm just showing 4 examples here, meaning there really is no role of inversions in suppression of recombination and sex linkage in this species. But we had a hint that coverage or sequence content was playing a role. And so we brought in those 200 samples that we had phenotyped for sex. And what was really interesting to see is, just like we were hinted at with our comparative genomic approaches, is that males tend to have about average coverage in this region, actually quite a little bit less than 1 on average, but females tend to lack any sequence content in this region on average. So it confirms this role of copy number variation playing a role in sex determination in water hemp. But again, it's not that simple. What we were really shocked to find was that there was about 10% of genotype to phenotype mismatch. So we found about 15 males for which they had pretty much no sequence content in this region. So they were genotypically female in this region, and about 5 females that were genotypically male. And those are highlighted in pink and purple, respectively. So you can see that both on sequence content and based on a PCA of genotypes in this region, that clearly must be reflecting some mode of ongoing gene exchange between the X and Y. And so this could be gene conversion, this could be recombination. One approach we've taken to look at this was just investigating kind of the effective recombination rate in this sex-linked region. And what was kind of surprising to see is right in the middle of that region, we actually see a peak of inferred effective recombination rate, meaning that might actually imply that indeed, despite potentially sex-linked alleles and sexually antagonistic alleles accumulating on this, there's still lots of ongoing gene exchange, which is not what you would expect. Finally, we see both that sequence content and haplotype diversity in the Y is really strongly shaped by evolutionary history. So there's two ancestral lineages in Amaranthus tuberculatus, and that significantly explains the amount of sequence content present in both males and females in this region, but also genotypic diversity. And so despite the fact that we often think of Ys as these gene depauperate regions, that's really not the case with what we're seeing here and what we think now to be this ongoing, sex-determining region with really ongoing evolution. So I think this is a really interesting, if not messy, system of sex determination in a kind of a recently evolved system. We see considerable diversity in these sex-linked haplotypes shaped by ancestry. What I didn't also talk about is it's also shaped by latitude, so climate adaptation might be playing a role in the diversity we're seeing there. But in terms of this two-step or quantitative selection model, I think we actually might be somewhere in between. So we've identified a large effect locus, but we clearly see that there's ongoing recombination. So we haven't completed kind of that two-step model. I think that in order to explain kind of those genotype to phenotype switches, that we're going to have to invoke an additional modifier contributor to sex. And that could be environmental, or it could be other loci across the genome that might be encoding sex in those individuals. So I think this is pretty cool because there's some really interesting recent models about how gradual selection for Daiichi can actually occur through sex allocation. And there's a really nice paper that I'd plug you to read if you're interested in this. So thank you guys for listening, and happy to take any questions if you have it.
Greetings, everyone. Thank you all for attending. My name is Audrey Portal, and I'm going to present to you the ongoing work of my first year of PhD, which aims to find out if the host-parasitoid relationship is a driver of horizontal gene transfer. So, evolution is traditionally represented through trees that illustrate the vertical transfer of inheritable traits from parents to their offspring. But the transfer of genetic material can also occur across the branches of the tree, between the lineages and beyond the breeding barriers. And this phenomenon is known as horizontal transfer, HT for short. Now, horizontal transfer is known to be prevalent among prokaryotes as their main mechanism of genes acquisition, but it also occurs in organisms in which the germline is isolated, such as metazoans, and in my work, I'm focusing on several orders of insects. Although horizontal transfer is increasingly well known among insects, there are still several questions that remain to be answered, such as the prevalence of the phenomenon and the ecological and molecular mechanisms that are involved. One frequent hypothesis in the literature is that intimate ecological relationships could promote horizontal transfer events because of the durable physical proximity that they induce between the organisms that are involved. And in my work, I'm focusing on a specific ecological relationship, that is the host-parasitoid relationship, quite prevalent among insects, with the following hypothesis that the host-parasitoid relationship could promote horizontal gene transfer events among insects. Now, some of you may be wondering, what is a parasitoid? So parasitoids are organisms that are going to lay their eggs inside a living host. So in the left, you have a picture of a Hymenoptera, so a wasp parasitoid, and on the right, a picture of a Diptera, so a fly parasitoid, both laying their eggs in Lepidoptera larvae. And so the progeny of the parasitoid is going to develop inside the host, often killing it at the emergence, quite like the alien of the eponymous movie that they actually inspired. But this ecological hypothesis for horizontal transfer is actually backed by a molecular hypothesis. You see, some Hymenoptera parasitoids produce what we call polydNA viruses that, despite their name, are not viruses per se, but domesticated viral particles that are produced by the wasp. So they look like viruses, but they contain wasp genes, and these are injected in the host while the peristoid is laying its eggs. They can integrate wasp genes into the host cells, and these are going to inhibit the host immune system, allowing the progeny of the peristoid to develop free of trouble. So these wasps use quite an amazing natural transgenesis mechanism that could be a mechanism, a molecular mechanism for horizontal transfer. And several cases of horizontal transfers that have actually been mediated by these particles have been identified in the literature. Note that as of yet, no analogous mechanism has been identified in Diptera peristoids. So, as you can see, the combination of ecological and molecular hypotheses for horizontal transfer makes the host peristoid model quite ideal for the study of horizontal transfers and its underlying mechanisms. And how to tackle this hypothesis within an extensive enough data set. So the data set that I use in my work is derived for more than 40 years of fieldwork from Daniel Jensen and a huge team of fieldworkers into the Área de Conservación Guanacaste, a natural reserve located in the northwest of Costa Rica, where they extensively documented all ecological connections between thousands of local insects, and the DNA samples were accessible thanks to the barcoding efforts conducted in the University of Guelph and directed by Paul Hebert. So the data set that I actually use is 222 genomes of selected Lepidoptera hosts and their Hymenoptera and Diptera peristoids that constitute an exhaustive ecological network in a well-known phylogenetic frame in which my work was to identify horizontal transfer events, we'll see how later, and to determine the effect of the host-peristoid relationship on the frequency of horizontal transfer events. Now, to quantify this effect, we had to be wary of the problem of phylogenetic inertia. That is, when looking for correlations in vast phylogenies, some observations can be not independent because of phylogenetic closeness. And to avoid this, we designed a paired approach among distinct clades to only focus on independent observations. It goes as follows. So first we take a pair of species, a peristoid and a host, that are connected by the host-peristoid relationship. And we take a second pair of species, another peristoid and the same host, but they're not connected by the host-peristoid relationship. We'll call this one NP for non-peristoid of H. And so here we have a group of 3 species that we call a triplet that is comprised of 2 phylogenetically comparable pairs because P and NP are close species located in the same clade. And the idea here is to multiply such observations across independent clades to be able to rigorously quantify the effect of the host-peristoid relationship on the frequency of horizontal transfer events that we will count in each type of pair of each triplet. And this way we obtain 48 such independent clades. So now that we have a good framework, we still have to detect horizontal transfer events. This was done by using a genetic distance-based method. So here's the idea. You have here the tree of the 3 species of the triplet. They have diverged for a certain time and thus are separated by a certain genetic divergence. Now, if we have an event of horizontal transfer between P and H, the homologous region that results is going to be way less divergent than the overall expected divergence between the lineages. And this is the horizontal transfer signal that I was looking for in my genomes. In practice, I first used a genes prediction step to only focus on the protein coding sequences of my genomes. Then I regrouped all resembling sequences with a clustering step with a 90% identity threshold to really get this low divergence signal of horizontal transfer. And I also, to avoid any contaminations, I eliminated all clusters that exhibited identical sequences between a host and a peristoid species. And the remaining clusters were all the signal of one horizontal transfer event. So we could count such events for each type of pair of each triplet. And the statistics of the statistical test of the hypothesis was the mean divergence of horizontal transfers that occurred between the species that are connected by the host-peristoid relationship and the ones that they are not. And so under our hypothesis, we would expect this value to be positive. So let's see what we got. We found a total of 434 horizontal transfer events scattered across our 48 independent clades, and a mean number of horizontal transfers per host per histode per clade of 2.08, which is not a very strong signal, but we can already see that there seems to be an excess of horizontal transfers between the species that are connected by the ecological relationship. And this tendency was confirmed by a statistical test. So here you have the distribution of the null hypothesis. The vertical red line is the value of our statistic, and we can see that it's positive, located on the right of the distribution. So there seems to be a mild effect of the host per histode relationship on the frequency of histode transfer events in our insects. But remember that our histode transfer events involved Hymenoptera and Diptera parasistoids, two separate orders. And so we wondered what will happen if we did the test on the separate subsets. So on the subset of histode transfers involving only Diptera parasitoids, we found a mean number of histode transfers per host per histode per clade of only 1 per 33. And we can see that the value of our statistic is again positive, but even more off-center from the distribution of the null hypothesis. So there seemed to be here a strong effect of the host per histode relationship involving Diptera parasitoids on the frequency of histode transfer events that they share with their hosts. On the subset involving only Hymenoptera parasitoids, we found a mean number of histode transfers per host per histode per clade of 2.49, so almost a double of what we see with Diptera parasitism, which means that they transfer almost twice as much with their host than what Diptera do. But there's absolutely no effect of the host per histode relationship involving Hymenoptera parasitoids on the frequency of histode transfer events that they share with their host, which was quite a big surprise to us. Remember that Hymenoptera do have a molecular mechanism for histode transfer. It's still a hypothesis, of course. So we were expecting a strong effect of the host per histode relationship involving Hymenoptera on the frequency of histode transfers, which is not what we see. The fact that they transfer almost twice as much with their hosts than what Diptera do per clade suggests that on average, Hymenoptera do have more opportunities for histode transfer, probably thanks to their molecular mechanism, but they transfer equally with the host that they parasitize than with the host that they do not. One possible explanation for this could lie with the assessment of the host per histode relationship. So this was done by observing what emerged from Lepidoptera cocoons. So for each host species, out of more than 10 observations, if a parasitoid emerged at least 5 times, it was labeled as a parasitoid of the host, and if it never emerged, it was labeled as a non-parasitoid of the host. And this method could have missed rare attack patterns from parasitoids labeled as the non-parasitoids of the host, that even if unsuccessful in letting the Lepidoptera emerge, could have mediated histode transfer events because of how their molecular mechanism works. Another possible explanation could be that host per histode relationship changed recently. So a histode transfer that occurred between a host and its non-parasitoid could have happened while the species was effectively a parasitoid of the host. In this case, the discrepancy of effect between Hymenoptera and Diptera parasitism could be partly explained by a greater stability of the host per histode relationship between Diptera and their hosts than between Hymenoptera and their hosts. One last word about the molecular mechanism. Keep in mind that there could be a molecular mechanism that has yet to be discovered in Diptera, and finding traces of such a mechanism is actually a huge part of the PhD studies of my colleague Sarah Ukal. So if you haven't seen her talk this morning and are interested in the subject, I strongly advise you to go talk to her. And I would like to precise that the results I presented here are only preliminary. Several steps of improvement are already ongoing, mainly to find more histode transfer events to strengthen the signal. So this is only the beginning. I would like to thank you all for your attention, thank my marvelous colleagues that helped me do all this work and that will help me in the future. And I would be happy to take any questions. Yes?
Yes, that's some really intriguing work. With horizontal gene transfer, I do always wonder about contamination. You said these are samples directly from the field, not lab-reared.
these samples are, yes, actually from the field. They were processed, of course, by people who are not me. I'm only working on all the process stuff. And we are well aware that with such a subject, contaminations could be a huge problem. So as I said, one of the measures we took against it was to get rid of all identical sequences that we found.
Were there lots of those?
There were some, not that many. I didn't mention it, but I also did a taxonomic analysis of all the candidates for horizontal transfer that I found. And I found many to be from bacteria assignated. So these might be a huge part of contaminations, and we're still looking at processing all the genetic material that I have more to avoid this. But these statistics are actually done on the horizontal transfer candidates that are not assigned bacteria. Yes?
Yes. Are there particular genes which are more commonly found in these transactions? I mean, because even because the process or because they're selected.
So I have done a preliminary functional assignation to what I found, and there are many, many, many proteins from transposable elements, which is not that much of a surprise, and further studies are going to elucidate if where they come from, and I'm also trying to find more matter. So, but we expect a lot of transposable elements. And I see that it's time for me to leave. So thank you all for your questions.